All right, if you were a sperm and Lucky was deposited into the heifer's tract via natural service by the bull, where would Lucky be deposited? All right, we'll go 15 more seconds here. Okay, guys, let's go ahead and wrap up those responses. I'm going to go ahead and lock them in. Okay, so we have a couple of options here. All right, so most of you are saying the vagina, some are saying the cervix, and some are saying the uterus. Okay, good. So I do want to talk through this a little bit. Oops, sorry, that was my bad. All right, so some first things to notice here in this question, okay? Again, guys, this is a 300 level class. I'm asking you to look at things in more detail, and that includes even quiz questions, okay? So if you were deposited into the heifer's tract, that's your first clue. Your second clue is using natural service, okay? So that indicates that the bull is actually breeding the cow. We're not using artificial insemination. We would be deposited in the vagina. Okay, so and more specifically the fornix vagina, but I didn't get that specific in the answers. So vagina is correct here. All right, so let me switch gears just a second. And go to all right. So with where is my annotate? There we go. Okay, so with a heifer, okay, so the sperm cells are actually going to be deposited right here. So this is going to be the fornix end of the vagina. Okay, so with the heifer, that is where the bull is going to deposit the sperm cells in a natural setting. All right, if we were doing artificial insemination in this heifer, like we're going to talk about a little bit later in chapter 7, we would actually be depositing semen, not through natural service, but through artificial insemination into the uterine body. All right, so the whole point of artificial insemination is to increase the chances of the sperm cell actually getting to the ovary or to the oviduct for fertilization to take place. All right, so that sperm cell is going to need to be deposited here in the uterus, and we can do that by using an artificial insemination gun. Go in through the vagina, work our way through the cervix, and just on the other side of the cervix, here in this blue area, the uterine body, is where we would deposit the semen in an artificial insemination scenario. All right, but this question is asking, where would the sperm cell be deposited via natural service? And so again, that is back here in the fornix vagina before the cervix. Okay, so let me stop sharing really quickly. What questions can I answer for you about that concept? I want to make sure that we are very clear on the difference, not only between species, and we're going to look at a different species here in just a second, but also between natural service and artificial insemination. Questions?
Casey, I just had a question. So like with the fornix vagina versus just the vagina in general, is like the vagina referring to the entire area from the vaginal opening up to the cervix, and then the fornix vagina is just more specifically right in front of the cervix?
Yes. So good
Okay.
question, Katie. So the entire vaginal cavity is pretty big, right? And so this whole structure outlined here in red is considered the vagina. The reason that we get as specific as we do about the fornix vagina, which is going to be up next to the beginning of the cervix, is because what can actually end up happening is here in this area and in this area, we can get into blind pockets. Okay, and so in an artificial insemination scenario, if we don't have a good idea of what the cervix feels like and us being able to work the artificial insemination gun through the cervix, we could actually deposit the semen here in the fornix vagina, thinking we're in the uterus. Okay, so that is one of the big reasons that we are being distinctive between the vagina as the whole and the fornix vagina, is because more often than not, just because of the length of the male's penis or the ability to use the artificial insemination gun, we can actually dump semen into the fornix vagina, or this end of it. And that is going to increase our chances of getting more sperm cells through to actually being usable for fertilization, right? If we dump semen clear back here in the backside of the vagina, there's a really good chance that some of that sperm material could actually go back out of the cow. Okay, because remember, she's going to be in a standing heat most likely in this scenario. So there's going to be lots of contractions happening that is going to push that vaginal wall around, and it can actually push semen back out through the vulva. So that's why we want to place it both in the bull scenario closer to the cervix. So does that help answer that question, Katie? Okay, great. Any other questions on the bovine placement via natural service of the sperm cells? Okay. I don't think there's anything in the chat. Let me stop. No. All right, let's go back to poll everywhere. Okay. All right, so our next question, let me get it activated, is if you were a sperm cell and you're going to be named Lucky again, and you were deposited into the gilt tract via natural service by the boar, where would you be deposited? All right, so similar concept, different species. Okay, we're going to take 30 more seconds here. All right, go ahead and lock those answers in. See what our responses are. Okay, so I have some people saying the vagina, a lot of people saying the cervix, and some people saying the uterus. Okay, if you answered the cervix, you are correct. All right, so in the sow's reproductive tract, or in this case, the gilt's reproductive tract, we are, come on, dude, where is my annotate? There we are. All right, so the vagina is much shorter than in the cow's reproductive system, okay, but the cervix is much longer. What is another big difference between the cow, or between the heifer and the gilt's cervix, and another anatomical difference, and just shout it out because I can't see the chat right now.
The sows have like a corkscrew kind of deal.
They do. They have a corkscrew configuration. Okay. And then the cows have annular rings. So that corkscrew configuration in the sow was created to complement the boar's penis structure. Okay, so at the end of the male or of the boar's penis, it actually has a flagellum that looks like a corkscrew. So it actually twists around. And so the whole reason for that is so that the penis can actually lock in to the cervix here and deposit the semen. All right, so in the scenario for sows or gilts, the semen placement through natural service is going to be in the cervix. All right. So if you answered cervix on this question, you got it. Okay, if you answered another anatomical structure, we need to go back and review those. All right. So with that, what questions can I answer for you about the difference between bovine and the sow or gilt, ovine and porcine? That's what it was, the word I was looking for. Anatomical structure.
Just to clarify, the reason that it goes all the way in the cervix, it's because of the concentration of the semen, right? Because I think in the notes it was that the boar has the highest amount, but has the lowest like actual semen count.
Yes. So,
something like that.
Yeah, so there's lots of reasons that the system was designed so that the boar's penis actually locked into the sow's cervix. Okay, so first of all, one of the reasons is that sows or pigs in general are a litter-bearing species, right? So they're going to have multiple piglets per horn. So that's one reason. A second reason is exactly what you said, Daphne, is that the boars have the highest volume, but the lowest concentration of sperm cells. Okay, so we have to, because they're a litter-bearing species, you have to, one of the reasons that system was designed is to increase the chances of getting the actual sperm cells to the oviduct for fertilization.
Okay.
Right. So all of those things are complementary.
Thank you.
Yep, you bet. Good questions, guys. Any others about female anatomy? Okay, great. All right, let's go back to poll everywhere. All right, so thinking about last week's material, this is where you guys can actually text in or respond in with a response. So first of all, I want to know, how did last week go with chapter 3 material? And then what was one thing that you learned? And if you need a reminder, chapter 3 was male anatomy. Okay, good. Differences between penis structures in different species. Yes, a lot of new repro tracks to remember. Most of them have the same anatomy and structure though, okay? So they just look a little bit different, but the order is going to be the same. Thermoregulation, okay, good, 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 good. A little overwhelming. Excellent. Lots of terminology. Yep, these are all chapters that if you guys are flashcard type learners, that would be good to start creating flashcards. Okay, male anatomy, good, good, good. Excellent. Okay, let's take 15 more seconds here. If you guys haven't got your answers in, let's do that. Okay, good. Excellent. Okay, let's go ahead and stop. I'm going to lock these answers in. Thank you guys for participating. It looks like a lot of the stuff that was kind of the big objectives is what you put in your response. So that's great. Let me look at something real quick. Okay. I'm going to go ahead and activate another question for you guys. Okay, so scrotal temperature needs to be blank degrees blank below body temperature to ensure sperm viability. So this question is asking for two pieces of information, the number of degrees and then the unit of measurement of those degrees. So Celsius is abbreviated as C, Fahrenheit is abbreviated as F. All right, so we'll take about 45 seconds to get these answers in. All right, 10 more seconds here. All right, let's go ahead and lock these in. Okay. Most of us got it. Great. Okay, so 5 degrees below body temperature. Okay, so 5 degrees Celsius below body temperature. That is our window. So anything warmer than that is going to adversely affect sperm cells. Anything cooler than that is going to also adversely affect sperm cells. Okay, so let's talk really quickly about chapter 3. All right, so chapter 3 again was another big chapter. And so let's get this. There we go. All right, so we started off the chapter talking about general anatomical structure. So you guys saw a series of cartoon type slides like this one, followed up by a live repro track slide like this one. And so this first part of the lecture was all about making sure that you got down anatomical structures of the male repro system. And I'm hoping a lot of this was just a review of what you guys had learned in your respective Animal Science 101 classes. All right, so the pictures of the actual tracks look a lot different than the cartoon, as they always do, but you can pick out the different anatomical structures here, and they're abbreviated. These are all out of the Singer textbook. Okay, and so the general path of the sperm cell is going to start here in the testes. That's where spermatogenesis actually takes place. We're going to get more into depth with that concept in chapter 10 later on in the semester. But what we actually end up having is these lobules here are going to separate out the base membrane from the areas where the sperm cells actually will grow and develop into correct sperm cells with a head, a midsection, and a tail. And then those sperm cells will then move into the mediastinum through the seminiferous tubules, which is the spaghetti looking like structures here in these lobules. And they're going to travel here into the mediastinum and the root tubules. And this is kind of the center part of the testes. And so then those sperm cells will travel from the mediastinum up into the head of the epididymis, into the body of the epididymis, where they become functional and mature, their last final step. Then they'll move into the tail of the epididymis, which is going to be an area for semen storage. Once they are recruited out of the tail of the epididymis, they will then head through the rest of the reproductive tract, and their end goal is ejaculation out of the glans penis. All right, so the sperm cells, once they are recruited to move from storage in the tail, they will then move through the ductus deferens here up into the body cavity. All right, so once we are up into the body cavity, the sperm cells will move through, the ductus deferens, and then into the tubule system, past the ampulla, which sits on top of the uterine body here, and into these other secondary accessory glands, where they will be, or where the sperm cells will be volumized with different seminiferous fluids, and that work as buffers, protection, all of that good stuff. And then the sperm cells will work down through down through the rest of the penis. The penis will extend, and then, and then the sperm cells will move out through the glans penis. And so that is the general track that the sperm cells will take. So spermatogenesis, the process of generating sperm cells, happen in the testes. Storage or maturation and then storage happens in the epididymis, and then the rest of the highway, if you will, for the sperm cells goes back up through the body cavity. Fluid is added to protect the sperm cells, and then it will go out. Those sperm cells will go out the penis and finally out the glans penis. And so that was the first part of the lecture last week, was just getting the anatomical structures down. So, and you guys saw that with the worksheet as well, to where you got to actually go through and label different areas of the anatomical structure. The second half, or the second part of the chapter 3 lecture last week really looked more in depth at each piece of the anatomical structures. And so I'm not going to go through all of them today. I just want to hit a couple of them that tend to be a little bit confusing, and I want to offer you guys the chance to ask some questions here. So in the testes, we essentially have two areas in the testes that are separated by the basement membrane. And so that basement membrane, if I can get my hands, or my, come on, go back. Where is, come on, buddy. All right. looks like it doesn't want to work. So we'll look at this bigger picture here. So this basement membrane here separates the interstitial compartment from the basal compartment in the adluminal area. And so it's really essential that these two compartments in the lobes of the testes are separated, because the cells that are found in the seminiferous tubule, so in the top part of this photo here, that is made up of the basal compartment in the adluminal area, these are where we're actually going to find the germ plasma of the male, okay, so, or spermatogonia. And so the spermatogonia is going to start as a Sertoli cell. So you can see these little itty bitty black cells down here. They are those Sertoli cells are going to be analogous to granulosa cells in the female or in the follicle. Okay, so they actually are packing the DNA material of the male. And so the Sertoli cells in this luminal compartment are going to help govern spermatogenesis. Okay, the Sertoli cells are going to be receptors for not only follicle stimulating hormone in the male and testosterone as well, but it's also going to produce androgen binding protein, or ABP. And this is a really essential protein for transportation. Okay, so if that protein is not present, then the sperm cells will not mature correctly. The Sertoli cells also are going to secrete inhibin. Okay, so inhibin is a hormone that will help suppress follicle stimulating hormone coming from the anterior pituitary. And so inhibin is really important because it provides a negative feedback mechanism so that the sperm cell production doesn't get unregulated and out of control. All right, and then the Sertoli cell will also assist in the formation of the blood-testes barrier. And so that Sertoli cell, again, is going to be in this adluminal compartment where the germ cells are also present. All right, if we look outside of the seminiferous tubules, okay, so the lower compartment here, the interstitial compartment, this is where the Leydig cells are actually produced. And so these right here are the Leydig cells. I'm hoping you're seeing my cursor because my annotation's not working right now. And so these Leydig cells produced the steroid testosterone. And testosterone, again, is essential for driving the maturation of the germ cell into a sperm cell that looks like a sperm cell. So we can kind of see that up here at the top. Okay, so we have these spermatocytes that are going to be kind of globular or round in configuration. And then as they move towards the adluminal area here, they slowly start to morph into what we really recognize as a sperm cell, with a head piece, a mid piece, and a tail piece. And so that maturation process is driven by testosterone actually being the regulator of spermatogenesis. All right, so it's really important. I just wanted to take a second to make sure that you guys understood what was going on in the lobes of the testes. And that's where we see two areas of that are really equally important. They work together, but they're separated. And they're separated by that basement membrane. On the top side with the germ plasma is going to be the Sertoli cells. On the bottom side without the germ cells is going to be the Leydig cells that produce testosterone. All right, and then towards the end of chapter 3, we started looking at different characteristics in penis structure among different species. So this slide just kind of sums up what some of the big differences are. And so you guys had some questions on the worksheet about that, like the difference between a bull and a boar penis, okay, those type of things. So a bull penis is going to have a fibroelastic structure, meaning that the hormone relaxin is actually has to be upregulated for that penis to elongate. And so that fibroelastic ligament to actually relax so the penis can elongate. The bull also has a sigmoid flexure type penis, as does as does the ram. So that sigmoid flexure just means that the penis has the ability to relax and contract, okay, in a sigmoid structure or an S structure. The difference that we see between the bull and the ram penis is even though the ram also has a fibroelastic penis, at the end of the glans penis that will actually enter into the female reproductive tract is a filiform appendage. So it's actually just a piece of tissue that can range anywhere from probably 1/2 inch clear to 1.5 inches in length, depending on genetics, age of the ram, etc. And that filiform appendage is really essential because there's actually a flap on the end of the ewe's cervix near the fornix vagina that filiform appendage actually has nerve cells associated with it and can flail around and open that flap, allowing semen to get into the cervix. So we've had a lot of difficulty actually confirming that is what that appendage does. So that is what we think is happening in the system at that point in time, but it's really hard to create enough research to confirm that. So that's what we think is happening, but we aren't 100% sure as scientists at this point in time. And then another major difference between the bull and the ram is that if you guys looked at the testicular size of a ram compared to a bull, they're pretty comparable. All right, so meaning that that even though the bull is a bigger animal, his testes are about the same size as a ram. So that means that the ram has as much surface area and testicular area to produce semen. Therefore, ram semen tends to be small in overall volume, but very high in concentration. So basically the opposite of the boar. All right, and then the boar penis is quite a bit different, and we've already talked a little bit about this. So first of all, the boar actually is going to produce an odor. Okay, so if you guys have been in and around the swine unit when breeding is taking place, the boar is definitely going to smell a little funkier than other species, except boar goats. Oh my gosh, they stink really bad. And then in the boar, the testes are actually inverted. Okay, so they cross over inside the body cavity there. And so the boar's testes are going to be outside of the body cavity, but attached to a higher degree than the pendulum that the bull and the ram has. And so the boar, and so that is another major difference between the bull and the ram compared to the boar as far as testes differences. And so we also see that the penis in the boar has that corkscrew configuration in the glans penis that allows it to actually lock into the sow or the gilt's cervical tract for deposition of the semen. And then, like we just mentioned, the semen in the boar is very high in volume, but low in concentration, the opposite of the ram. The boar also has a sigmoid flexure type penis and has a very large bulbourethral glands. So the gland structure helps with creating the additional volume that is going to go into the ejaculate. And then we also talked a little bit in the lecture about the big difference in the stallion as well. So they have a completely different type of penis. They have a vascular penis and not necessarily a fibroelastic penis. All right, and then as we got to the end of the chapter, or end of chapter 3, we talked about thermoregulation of the testes. So why it is so important that the testes be maintained at 5 degrees Celsius below the body temperature of the animal. And so thermoregulation is actually regulated about 5 different ways. So it's regulated through scrotal control, through the temperature difference, the body temperature versus the temperature in the testes. The muscles will help regulate thermoregulation as well. Respiratory effects can help regulate the temperature, and the heat effects can have really damaging issues or damaging effects on sperm cells. And so thermoregulation is really, really essential in the males. And that's one thing that we have to watch and we can help manage for as livestock managers. All right, so let me stop really quickly. I know we're getting kind of short on time, and I wanted to dive into embryogenesis just very quickly, but I want to take a minute. Does anybody have any questions about chapter 3, male anatomy?
I don't know if the question makes any sense, but since the male body is, so to say, there's a lot more thermoregulation going on than in the female, would it be safe to assume that male animals are more susceptible to things like heat stroke than female animals?
Interesting question. So I don't think they're necessarily more prone to heat stroke, but they are definitely more prone to heat having adverse effects on sperm cells. So yeah, so male and female animals can handle heat very well. And we've developed things through time or through the evolution process to deal with that, right? So we have certain breeds that do better in hot temperatures, certain breeds that do better in colder temperatures, that type of thing. But as far as the effects on the sperm, that's what we're really talking about with thermoregulation. So yeah, if it is super hot outside or even super cold, and the males actually will engage the muscles and the cremaster muscle to pull the testes up next to the body cavity when it's cold. If it stays up there too long, it will be sequestered, or will be exposed to higher body temperatures, and you tend to get less viable sperm in those situations. So you can have infertility in cold weather as much as you can have infertility in males in warm weather.
Oh, gotcha. That makes sense. Thank you.
Yeah, you bet. Good question. All right. Any other questions about male anatomy? All right, great. I'm going to go ahead and we're just going to try to work through a couple of slides about chapter 4, which you guys are getting into this week. So a lot of this will make more sense as you get through this week's lectures. All right, so with embryogenesis, what you guys need to kind of keep in the front of your mind, by the time you get through this chapter, you should have a really good understanding of the primary embryonic germ layers. You should understand the pituitary development. You should be able to understand and communicate that sexual differentiation of the reproductive tract actually involves very specific substances, okay? Specifically proteins that are the product of gene regulation. And then the last major objective is to be able to understand and communicate the regulation of sexual differentiation in the male and the female. All right, so as we start looking at the embryogenesis, when conception takes place or fertilization takes place, we are going to get a mass reproduction of cells. And as those cells start to replicate over and over and over again in the embryo, it's going to create a structure that looks similar to this, and to where certain cells are going to arrange themselves along the outside. That is known as the ectoderm. And those cells will eventually turn into and differentiate into the nervous system as well as hair and skin. Okay, the second major structure here is the blastule. And these cells here will differentiate into the digestive system, the lungs, and the endocrine system. And then finally, the mesoderm, or the intercellular mass, which is what we also call it, is going to differentiate into muscles, the skeletal structure, the cardiovascular system, as well as the reproductive system. So the reproductive tract and the pituitary and the hypothalamus are all going to come out of the mesoderm and the endoderm. All right. So another big piece to chapter 4 is these sex determination. So how do these embryos that start clear back here as a big mass of cells turn into their specific sex? And it actually comes down to the presence or the absence of the X or the Y chromosome. So keep in mind that the chromosomal structure is actually going to determine the sex of the individual from a primary standpoint. So if we have 2 X chromosomes or a female, then we do not have a Y chromosome. And as obvious as that might be, the reason that I state that is because the Y chromosome is packing a gene here called the SRY gene. Okay, and so the sex determining region on the Y chromosome is what that stands for. So SRY gene. So if there is only 2 chromosomes present, there's an absence of the SRY gene and subsequently the SRY protein. Therefore, the default system is that that embryo will develop into a female. However, if we have an X and a Y chromosome, like in this configuration on this figure here, and the SRY gene is present and functional and not mutated, then the SRY gene is going to go through transcription and translation and produce the SRY protein. And so the SRY protein is then going to result in the testes being developed and that testes determining factor protein being produced. So once the testes are developed and that protein is produced, we will then see Sertoli cells start to secrete AMH, or anti-Müllerian hormone, and dihydrotestosterones, or DHT. And so once AMH and DHT are secreted from the Sertoli cells in the testes, then we see an upregulation of DHH, which is a gene that causes differentiation or development of Leydig cells in the male. Those Leydig cells will produce testosterone, and testosterone will result in the development of the male ductal system. Right, so the epididymis, the ductus deferens, the secondary sex glands, and the penis. All right. In addition, the Sertoli cells' production of AMH and DHT will also cause testosterone to degenerate the paromesopharynx duct, and that you might just put a circle around that. That's going to come up in the fuller lecture, okay? I'm not going to get into it today just from a time standpoint. And so once we see those paromesopharynx ducts go away, then that allows more dihydrotestosterone to be produced, and we see the development of the penis, the scrotum, and the secondary sex glands. So the take-home message here is that if we have an X and a Y chromosome, that Y chromosome is going to have the SRY gene on it, if that SRY gene is present and produces the protein, we're going to see a male develop from that embryo. If there is 2 X chromosomes and no SRY protein, by default, the female anatomical structure will occur. All right, so that's sexual differentiation. And then you will also see, I'm not going to get into it today because I think we're getting tight on time. People are leaving, heading to their next class. You're also going to see a discussion about the descent of the testes, and some conditions that we see in livestock, both the inguinal hernias in males and free martins in females. And so that will be at the end of the chapter 4 lectures. So that is what you can look for this week as well. And so I just saw something pop up. Let me check Blackboard really quickly. Okay, and sorry, it looks like week 4 was not released on Friday, so I'm sorry about that. It should have been. It is released now, so you guys have full access to chapter 4 materials. So, all right, guys, that's all I have for today. We will meet again next week where we will review chapter 4 and we'll get into hormones, which is my favorite part of, or one of my favorite parts of reproduction. And so we're going to spend 2 weeks in chapter 5 of the Singer text or in endocrinology starting next week. So if you guys are having trouble, please get a bookings appointment with me. Come see me during office hours this week. All right, thanks, guys.